Welcome back to the second half of Chapter 7 podcast. We're going to start off here with eukaryotes and the structure of eukaryotes. So beginning here, the eukaryote itself just means true for the U part, and karyo just means nucleus. So these guys are named after the fact they have a nucleus. And that all comes down to the fact that a nucleus is one of the largest organelles, so it's one of the first things they noticed that some cells had and some cells didn't. Now the purpose of the nucleus is really to house and protect your genetic information more than anything else. And there's two general forms, if you will, that your genetic information can exist as. You have chromatin, which is essentially going to be loose DNA with some proteins, and then you've got chromosomes, which is going to be essentially condensed DNA with some proteins. When you actually look at a chromosome or a chromatin, it's actually going to be almost 50-50 proteins and DNA. So we like to think that it's all DNA there, but the DNA is wrapped around and stabilized by these proteins called histones. And in general, just like if you have your house, you're going to want your stuff loose most of the time so you can access it. But for the purposes of cell division, we're going to want to package things up, just like if you were moving. You'd package them up in these condensed chromosomes, so it makes it easier to get the DNA from one part to another, in this case to either end of the cell, so it can divide down the center. You'll also see in a nucleus that there'll be this darker spot called the nucleolus, and that's basically associated with making rRNA, which is one of the main constituents of what's going to be made, which is a ribosome. So we've got DNA, which has the code, specifically the code for proteins. That DNA will be used in the nucleus to make RNA, all the different types. Here you see the rRNA. There's also M and T we'll talk about later. And then those will ultimately be used to make proteins. So you will see that the nucleus has at least a very directly indirect role in making proteins. And then lastly, the nucleus is going to have an envelope, which is going to be this double membrane that surrounds the nucleus, and it's going to have these pores or gaps in it that will allow things like RNA to go into and out of the nucleus, while still giving you enough of a barrier that the nucleus stays pretty protected. Oops, let me jump down here. So the nucleus, if you look here, once again, you've got the outer and inner membrane. You can see here the pores. These are some ribosomes that are attached. You've got the nucleolus, so this would be helping make the ribosomes themselves. And then, I wouldn't worry about this, we'll get to this later. And then you've got, obviously, just various ways of getting in, out, producing stuff. And then we'll see that later there'll be the ER that's attached right to this that allows us to take care of what we need to. Ribosomes, you're going to see there's two types of ribosomes. Both of them are going to be dealing with protein synthesis. It's what they do. But there's two types, the free ones that are just going to be found in the cytoplasm, or the cytosol, if you will, and these are going to be the ones that make enzymes and proteins that are used in the cell. So they're typically going to be used in the cytoplasm. So this will be proteins for the cell. You also have the bound ribosomes, and those are going to be making proteins, but they're going to make proteins that are going to be for the plasma membrane to be embedded in it, or they're going to be exported out of the cell. So some of these ones will actually be, we'll just say, excreted, but they'll be released through the cell membrane, through the plasma membrane, into the outside part. So basically it's leaving the cell. This can be useful if you've got many proteins that make up this protein mesh that's on the outside of the cells that can hold cells together. You can also have certain proteins that grab specific nutrients, things that you need that you want to make sure you bring in. You'll also see that some of these that are excreted could be stuff like enzymes that break stuff down or ward off other stuff like bacteria. So there's lots of reasons you'd want to do this, but the whole point is these guys are going to the surface or beyond. These guys are going to stay in the cell. And we can jump through that one now. And so attached to our nucleus here, you can see that we're going to have the endoplasmic reticulum. That's that kind of folded structure we talked about before, and you can see the dots. That's going to be the rough stuff, and those will be the ribosomes we talk about, those bound ribosomes, whereas you have the other ones that are floating free. And this is going to be called the endomembrane system here that we're going to talk about, and that's going to be comprised of the ER, it's going to be comprised of the Golgi body, it's going to be comprised of lysosomes, vesicles, vacuoles, kind of all these things that just have a single membrane that surround them, but they're also used to transport things, where you can make stuff in the ER, you can transport it using vesicles, or store it in vacuoles, etc. It can then get processed by the Golgi apparatus, and then eventually excreted to the cell membrane or outside. So this is a very useful way that our cell's going to have that organizes, moves, and controls stuff. So ER, endoplasmic reticulum, this is going to be the stuff that's continuous with the nuclear envelope, as you saw there. It's going to have a lumen, which is the insides, and that will be largely used to transport stuff. So you can move stuff through the ER if you'd like. You'll also have two other types. Smooth ER is really just associated with lipid synthesis, so it can make lipids. For the most part, we're just going to leave it there. Rough ER, we've already said, is able to help make proteins. It'll also be involved in this transport. And you can also see that some things, as it's connected to the nuclear envelope, as it's kind of tied into stuff, some things will also use this just for actual support as attachment, so it can help us really compartmentalize, organize the cell, in some ways a lot like the cytoskeleton did. You know, when you see all these membranes, the whole purpose of these membranes is it lets you make organelles be essentially like a room, where you've got the walls that say, all right, just like we have outside the plasma membrane is outside the cell, and inside we have our own unique environment, that internal homeostatic environment. Now we can say we have our own unique environment inside the nucleus. That would be called the nucleoplasm. We have our own unique environment inside of the Golgi apparatus, inside of the ER. And so it'll be part of this compartmentalization that we see with eukaryotes, will be the other point that it does. So if I talk about ER, really I'm probably going to be talking about rough ER, because that does more of what we're going to talk about when we're dealing with ER. Okay, next we got the Golgi apparatus. If you remember back in middle school, they might have called this the UPS of the cell. And the basic idea here is it receives a bunch of stuff, typically from the ER. So things come from the endoplasmic reticulum, specifically usually the rough endoplasmic reticulum. It will then take and it'll modify them. So it'll add on these little oligosaccharides to make glycoproteins. It can add on those to lipids to make glycolipids, as they call them. And so it's going to go through and make these things that we need as markers, because those things will be important into identifying, is this supposed to be there or not, as our cells kind of go by and our immune cells scan our cells. They can look at the surface of our cell membrane for these trigger molecules. So it's going to kind of add all the stuff that we need so that our body can be safe before it ultimately is going to ship these things oftentimes to actually go through a transport vesicle to the outside of the cell, if we're excreting it, or once again it can be embedded in the actual cell membrane. So we can embed things in the membrane. So that's going to be its main job. And it will actually shift stuff, once again, it'll receive and ship stuff via these vesicles. So if you've got the Golgi apparatus, which for lack of artistic skill I'll just draw like that, you're going to have stuff coming from the ER in these transport vesicles, and then leaving via more transport vesicles to go to this outer membrane, where they can merge or they can kind of go outside. Cisternae, if you see this, that's just another fancy term for the actual like folds. So we said it's kind of like that stack of pancakes, so these would be called the cisternae, these various folds. I know it looks bad, but deal with it. Okay, next we've got lysosomes. Really, these are just a single membrane sac. There's nothing too elegant about them, but they've got hydrolytic enzymes, so they can break crap down. So they've got these enzymes in them, and this is useful because if we have something we want broken down, such as a bacteria that we ate, so you could have something like an immune cell that ate a bacteria, it would throw that bacteria into this lysosome or merge with a lysosome, and then those hydrolytic enzymes would cut it up and digest it to allow us to extract energy from it. You can also do autophagy, where things that break down, like maybe a mitochondria that's just not doing its job anymore, you can take and you can throw it in with a lysosome, you can get it busted down, you extract the energy, and you can reuse that energy elsewhere. Now some people have some hydrolytic enzymes that are missing or faulty, and so if you have some of these, you can get a metabolic disorder. These are some of the most dangerous ones. Some of them are treatable, where you just don't eat the stuff that's going to become toxic and hurt you. Phenylketonuria is one of those. But you have some of them like Tay-Sachs disease, where you're missing this thing that breaks down lipids that are commonly found within the brain. And so this lipid builds up in the brain, becomes toxic, and ultimately kills you at a very young age, like 4 or 5. So this is something that's pretty tragic, but it all comes down to not having the right hydrolytic enzymes within the lysosomes, so they can't break down the things that they need to naturally. Okay, vacuoles, these guys, in terms of looks, are going to be a lot like a big vesicle. They once again are a sac that has a single outside membrane, but their jobs usually tend to be a bit different. For instance, a lot of them will be used for storage. So you'll have where if you eat a bacteria, when you actually engulf it, so you kind of fold up over it and bring it in, you'll oftentimes have this bacteria now that's going to be sitting inside of this piece of the plasma membrane that you brought it in with, that you kind of engulfed it with. And so you can have this bacteria that's sitting inside of this, for lack of a better term, big vesicle, which we'll call a food vacuole. And so that would be one type of thing, storage. You have contractile vacuoles. These ones are used in some things like paramecia that live in conditions where there's water that constantly wants to come in. And we already said they have no cell wall. So if too much water comes in, you, for lack of a better term, explode. So they need to make sure they get rid of this water. So what they'll do is they'll dump this water into this contractile vacuole, which can then squeeze, and basically when it squeezes or contracts, it shoots the water out, which then keeps the paramecia from dying. And then central vacuoles is another type. This is the one that you see in plants. This is one of the identifying characteristics in plants, because when you see a plant cell, up to about 90% of it can be taken up by this big central vacuole. So it's even bigger, way bigger than the nucleus or anything else. All the other organelles just kind of sneak in here and chill around along the edges. And that one's going to be used largely for storage as well. There's some metabolic functions that occur there, but it's kind of like, I guess you could say, imagine if your house had a gigantic garage attached to it that you could use for storage. You could use it to get stuff done. You can use it to put waste there, or toxins in some cases, which can be used to protect yourself, both within the cell, so the toxins are isolated, compartmentalized, but also if something eats you and starts chewing on those cells, they can release the toxin, which should hopefully get them to stay away. And the tonoplast membrane, in case you see that, that's just the name for the membrane that surrounds the central vacuole. And so because in plants it plays a pretty big role in a lot of the stuff they do, you might see them talk about it. It's something that you will see frequently, just named, just saying, like, this is what's going on as you're shuffling stuff across the tonoplast for storage or bringing it out to use it. Okay, so that's basically it for the the endomembrane system. So stuff can essentially go from producing ribosomes and such in the nucleus to using the ribosomes on the rough ER, producing stuff which go via vesicles, etc., to the Golgi apparatus, and from there they can go out of the cell. And if you bring stuff into the cell, they can be essentially chewed up by our lysosomes, or they can be stored in our vacuoles. So all this stuff will be part of that endomembrane system. Now next we've got the mitochondria. And the mitochondria is kind of cool because it's one of the organelles that likely used to be a prokaryotic cell. And you can see that because it still has some DNA, you can see that here, and it does have some ribosomes. And so it's able to kind of do a lot of its own stuff. It even, when it divides, looks a lot like a bacteria when it divides. It does it in more of a binary fission type way, which is what bacteria will do. And you'll see it also has two membranes, which seems a lot like it was eaten, where this outer membrane seems more eukaryotic, and the inner membrane, called the cristae, seems a lot more like a prokaryotic membrane of an aerobic bacteria. Because what likely happened was an aerobic bacteria got eaten. All right? That aerobic bacteria was capable of using oxygen to generate ATP in a way that's more efficient than not. And so because of that, if the cell essentially forgot to eat it or decided not to, they can live together where that mitochondria can produce ATP and give some of it back to the host in a way that's more efficient than the host doing it itself, even if it only gets a cut, because you're talking 2 ATP versus 36 to 38. So it's pretty easy for it to kind of give you a kickback, and it's worth it. And then over time, what appears to have happened is that the mitochondria kind of got tamed, if you will, where a lot of the genes that it had transferred into the nucleus, so it's now controlled by the nucleus, and it doesn't act completely on its own. But you will see the ribosomes, the DNA, and within the mitochondria, the places that matter most is the matrix, which is going to be essentially the cytoplasm that's at the very center, the cristae, which is this inner membrane, and then this intermembrane space will play a role that's in between the inner and the outer. So we'll talk about how actual cell respiration occurs, but this will be the site of aerobic cell respiration. And so when I say that, I mean this is going to be the only type of respiration that needs oxygen. If you're not using oxygen, you do not need a mitochondria. It's not essential. There are also some bacteria that don't need mitochondria that still do aerobic respiration because they embed all these things that are in the inner membrane. They just embed them in the plasma membrane. The quantity will vary with cells. Cells that are very energetic, like muscles, you have lots of mitochondria, and cells that don't do much, like a skin cell, don't have many because they just don't need them. And we already talked about DNA ribosomes that helped us realize that they probably started off at their own cells. Chloroplast is another one that likely started off at some point as its own cell. This one is obviously found only in plants, algae, things like that are eukaryotic that do photosynthesis. They're also going to have two membranes. You've got an outer and an inner, except in these ones, neither of them do a whole lot. They have actually thylakoids, these membranous sacs that are inside. They're in these stacks. So each individual one like this is going to be a thylakoid. But the actual stacks of them, in case you hear them referred to, like this whole stack would be called a grana. At least that's how I pronounce it. And then this inner part, this cytoplasm, is ultimately going to be the stroma. So you can see that's going to be all this inner part. So the thylakoids and the stroma are going to be two of the critical pieces that allow things to do photosynthesis. The DNA ribosome part, it's once again similar to mitochondria. This is just useful because it indicates their origin, that these were likely cyanobacteria or photosynthesizing bacteria that also got ingested or moved into the cell and then started to kind of give back. And so they formed this symbiotic relationship until they eventually became an organelle. So these guys you see also have the double membrane, just like mitochondria. They have the DNA, they have the ribosomes. There are other plastids, that's the plast part. So there are some other things that we won't cover that you can find in plant cells that'll also be similar in terms of they've got DNA, ribosomes. They look like they were once their own cells, but they'll serve different functions than just photosynthesis. So if you see anything that's a plast, it typically means it's a plastid, that it's very similar to a chloroplast or a mitochondria in its likely origin, and it usually has a double membrane, has its own DNA, has its own ribosomes. Peroxisomes are a very difficult one. They have peroxide in them. Hydrogen peroxide is what you'll find. Now, hydrogen peroxide is not friendly stuff. This is the stuff you put on wounds that bubbles. You can use it in your mouth to just kind of shift it around, and then you'd have to spit it out because it will damage cells if you just kind of drank a whole bunch of it. But we use this because it can chew stuff up essentially. So we'll throw fatty acids and alcohol into a peroxisome and let the hydrogen peroxide break it down for us. And the nice thing is once this goes through and breaks stuff down, what it ends up becoming is water, and then it just has some oxygen gas that's left off. So this works out really nicely because we can use water, we can use oxygen gas. The only thing we have to make sure is that we don't let this stuff, the hydrogen peroxide, leak out. We've got to make sure that stays in these peroxisomes. But this is very useful as a, kind of like the lysosome, as a digestive organelle, if you will. So plant structures, as we just kind of wrap up here, we see the plasmodesmata. We've got those channels. That's something you'll only find within the plant cells that we'll talk about because animal cells don't have the cell walls, so they can't form the exact same thing, although they can do channels. You'll see that when you look at a plant cell, we'll have this large central vacuole, so that's going to help identify this. We've got chloroplasts, which you're going to find in plant-like cells. You will not find this in animal cells. We already brought up, if I can find where it's actually on here, the cell wall. So those are the three main things that you'll see that will identify it as being a plant cell, as opposed to if it was an animal cell, you would normally see centrioles. And the other thing that usually means it's an animal cell is lysosomes, because you're not normally going to be doing a whole lot of phagocytosis because you have a cell wall. You can't wrap around something because it's inflexible. And so normally they're not going to be eating much. So there are some plants that have this, but generally speaking, you will not have lysosomes if you're a plant cell. Those will be kind of unique to animal cells as we try and identify who's who. But most of the other stuff you can see they share, just like any eukaryote has. Eukaryotes as a whole are pretty similar. So while we like to kind of say plant cells and animal cells, in reality, they're pretty darn close to each other. So don't try and pretend they're so different. prokaryotes are actually much, much more different one versus another than any eukaryotes are. So while we look at eukaryotes and we give them all kinds of names, like there's fungus and there's protists and there's plants and animals, in reality they share a lot in common regardless of what they are. So we'll focus on these smaller differences, but don't lose sight of the fact that prokaryotes are actually a lot more different. I know people like to write them off as the small guys that are bacteria that can make you sick, but they're actually a lot cooler than people give them credit for. And then this is just the animal cells we wrap up, and you can see the ciliary flagella, which plant cells can have, especially certain cells like sperm, just like ours can. And so you'll see otherwise, though, there is no cell wall. You will see that there's not going to be any chloroplast. There's not going to be any central vacuole. The nucleus is typically the biggest guy that you're going to notice there. In this case, you will have lysosomes, although by sight, I don't think you'd be able to identify them that well because they look like most of the other membranous sacs. But that's going to be the stuff that just kind of works with this. You'll also see they tend to be much more fluid in their shape. Plant cells tend to be pressurized. Remember that cell wall, because they're filled with water, tends to push out against the cell wall. So they tend to have much more like solid shapes, I guess. Like they're going to be cylindrical or something that's going to be pretty rigid. Whereas with animal cells, you oftentimes see them drawn in a way where they're just kind of oozing about because they can. That cell membrane's still much more fluid because it's not surrounded by anything rigid. So it allows it to change shape more easily, and you can get more of shifting that goes on when you look at them.